Your hands have been keeping a secret your whole life. Two simple fingers, your index and your ring, hold the key to understanding something that science has been chasing for over a century. And now, finally, researchers have cracked the code. It's not about fortune-telling or ancient superstitions. It's about your DNA, your hormones, and a genetic mystery that reveals more about you than you ever imagined. For generations, people have looked at hands to make predictions. Palm readers claimed they could see your future written in the lines of your skin. Fortune tellers spoke about what the length of your fingers meant for your destiny. But what if the real story isn't in the creases and shadows? It's in the actual length of your fingers. What if that simple measurement could tell us something profound about the biology that makes you who you are? Not your future, but your past. Not your fortune, but your biology. Not mystical predictions, but scientific facts grounded in genetics and molecular biology. This is the story of how science finally unlocked the genetic code behind finger length. It's a story that begins in the 1800s with basic observation and reaches its climax in 2018 with one of the largest genetic studies ever conducted. Over 91,000 people participated in this landmark research. Researchers examined their DNA at 15 different locations across the human genome. And what they discovered changed everything we thought we knew about this seemingly trivial physical trait. Your fingers were telling a story all along, written in your DNA. Let's start with the basics. Take a moment right now and look at your hands. Place them flat in front of you. Now look at your index finger. That's your second finger, the one you use to point. Compare it to your ring finger, the one right next to it that you put rings on. Which one is longer, or are they about the same length? For most people, the ring finger is longer than the index finger. But for some people, it's the opposite. And for a select few, they're almost perfectly equal. This measurement is called the digit ratio, or more formally, the 2D to 4D ratio. The 2D refers to your second digit, your index finger. The 4D refers to your fourth digit, your ring finger. Scientists calculate it by taking the length of your index finger and dividing it by the length of your ring finger. It's a simple math problem based on a simple physical measurement, but the implications are anything but simple. The history of studying this ratio is longer than you might think, going back much further than most people would expect. In 1875, a researcher named Ecker first identified this measurement and noted that it seemed to differ between men and women in a systematic way. Decades passed with relatively little scientific interest in the subject. Then, in the early 1980s, a scientist named Wilson independently rediscovered this observation and began investigating it more systematically using modern research methods. But it wasn't until the late 1990s that this topic really exploded in scientific literature and captured the attention of the broader research community. A researcher named John Manning published groundbreaking work suggesting that the finger length ratio could serve as a biomarker, a physical indicator, of the hormones you were exposed to before you were even born during the critical period of fetal development in the womb. That's where things get really interesting. Manning's research suggested that the ratio was connected to prenatal testosterone and estrogen exposure. Before babies are born, developing fetuses are exposed to varying levels of sex hormones. These hormones don't just influence sexual development, they influence the development of virtually every system in the body, including the development of your hands and fingers. The theory went like this. If a fetus is exposed to higher levels of testosterone in the womb, their ring finger would grow longer relative to their index finger. The opposite would be true for exposure to higher levels of estrogen. This explained a curious pattern that scientists had observed. Men, on average, tend to have longer ring fingers compared to their index fingers. Women, on average, tend to have index and ring fingers that are much closer in length. Since male fetuses are typically exposed to higher levels of testosterone than female fetuses, this pattern made sense. But here's what makes it even more fascinating. There's tremendous variation within each group. Some women have distinctly longer ring fingers, some men have longer index fingers, and these differences seem to suggest different hormone exposure profiles during fetal development. But knowing that something exists and knowing why it exists are two completely different things. For decades, scientists knew that finger length ratio varied from person to person. They knew it seemed to correlate with sex hormones, but they didn't know the genetic basis. They didn't know which specific genes controlled this trait. That's where modern DNA science comes into play. In 2010, a major breakthrough occurred that would transform our understanding of this trait forever. Researchers conducted a genome-wide association study, commonly known as a GWAS, on children and teenagers from multiple populations. They meticulously measured the finger lengths of over 1,000 young people using standardized procedures and analyzed their complete DNA sequences. What they found was remarkable. A specific variant in a gene called Lin28B on chromosome 6 that was strongly and significantly associated with finger length ratio. The variant they identified had a designation of RS314277, referring to the EMA specific location on the chromosome. When researchers looked at the statistics, the p-value was incredibly small, 4.1 times 10 to the negative 8th power. In scientific terms, that's extraordinarily significant and meaningful. It's not a fluke. It's not random variation or noise in the data. It's a real, measurable, reproducible genetic effect. The Lin28B gene is involved in early limb development. It plays a crucial role in how your bones and tissues grow during fetal development. It's also important for stem cell development. Each copy of the minor allele of this variant was associated with a 0.6 increase in the mean 2D to 4D ratio. That might not sound like much, but in genetic studies, that's a substantial effect size. And here's the really important part. This finding was independently replicated. Researchers tested it again in a different population of over 3,600 children. The result held up. The same genetic variant showed the same effect. But the story didn't end there with just one gene identified. In 2013, other independent researchers conducting their own comprehensive GWAS found something different and equally important. They identified a gene called SMOC1 on chromosome 14 that was also independently associated with finger length ratio in their study population. The SMOC1 gene regulates bone growth and development throughout the body. It's hypothesized to control finger length growth directly by influencing how much bone tissue is laid down during development. Research indicated that one genotype of a single nucleotide polymorphism, or SNP, near SMOC1 was associated with a 0.8% increase in finger length ratio, while another genotype was associated with a 0.8% decrease in the ratio. That's an even larger individual effect size than what was found with the Lin28B variant. What makes SMOC1 particularly interesting is that its expression, whether the gene is active or turned off, appears to be controlled by testosterone and estrogen levels. This created an elegant biological model. The genes that control finger length growth are themselves regulated by the sex hormones you're exposed to in the womb. It's a cascade of genetic and hormonal influences that ultimately determines the length of your fingers. By 2018, the field was ready for the biggest and most ambitious study yet undertaken. Researchers from multiple prestigious institutions across the world came together to conduct a massive international meta-analysis of unprecedented scope. They looked at data from the discovery phase involving over 15,000 carefully selected individuals and a replication phase involving more than 75,000 additional participants from multiple independent cohorts. It was absolutely unprecedented in scope and scale for this particular trait. The findings were genuinely groundbreaking and represented a major leap forward in our understanding. They identified 9 entirely novel genetic loci, previously unknown to be associated with finger length ratio. 9 new locations scattered across the human genome where genetic variations had measurable influences on this trait. Combined with all the previous findings from earlier research, this meant that at least 15 different genetic markers spread across the entire human genome influence whether your ring finger or index finger will be longer. Nicole Warrington led this landmark study, which was published in the journal Human Molecular Genetics. The research team included geneticists from the University of Queensland, King's College London, the University of Bristol, Erasmus Medical Center in Rotterdam, and many other prestigious institutions worldwide. They had 23andMe's genetic data at their disposal. They had data from the Generation R study, tracking thousands of people from birth. They had data from the Twins UK study. The depth and breadth of the data was staggering. What they found reinforced what smaller studies had suggested, that approximately 60 to 70% of the variation in finger length ratio from one person to the next is due to genetics. That means your genes strongly determine whether your ring finger will be longer than your index finger. But it also means that genetics isn't the whole story. About 30 to 40% of the variation is due to environmental factors and other influences. Now, you might be wondering, what environmental factors could possibly affect finger length? The answer lies in the womb. The nutritional status of the mother during pregnancy can influence fetal development. Stress during pregnancy can alter hormone levels. Illness or infections can change the hormonal environment. These environmental factors interact with your genetic predisposition to create the exact finger length ratio you end up with. It's nature and nurture combining in the most fundamental way. But here's something crucial that scientists still don't fully understand: the relationship between the genes and the hormones. Do the genes control the hormones? Or do the hormones control the genes? Or is it bidirectional with constant feedback between genetic regulation and hormone levels? The evidence suggests that it's complicated. The SMOC1 gene's expression is clearly influenced by sex hormones, but the Lin28B gene's connection to sex hormones is less clear. This is an area where research is actively ongoing. The fact that genes are responsible for about 60% of finger length variation tells us that this isn't some random, meaningless trait. It's genuinely rooted in your biology. Your parents passed down specific genetic variants that influence finger length. If your mother had a longer ring finger, you're somewhat more likely to inherit the genetic variants that contribute to that pattern. Same with your father. Genes aren't destiny, but they're a powerful influence. Over the past few decades, scientists have become increasingly interested in what finger length ratio might tell us. If it's a marker of prenatal hormone exposure, might it correlate with other traits? Might it tell us something about health risks? Might it predict behavioral characteristics? The research has been fascinating, if sometimes controversial. One area that's been extensively studied is athletic performance and athletic success. Multiple independent studies have suggested that people with longer ring fingers, which indicates higher prenatal testosterone exposure, tend to perform better in various athletic endeavors and competitive sports. A recent 2024 study examined the specific relationship between finger length ratio and oxygen consumption, which is an important physiological indicator of cardiovascular fitness and aerobic capacity. The researchers carefully found that individuals with longer ring fingers metabolized oxygen more efficiently during standardized cardio testing protocols. This suggested a potential biological advantage in aerobic activities and endurance sports. But it's important to emphasize and note that this is just one small piece of the much larger athletic puzzle. Genes are never the whole story when it comes to athletic ability and success. Training intensity, proper nutrition, mental motivation, coaching quality, practice, and countless other environmental and behavioral factors matter tremendously in determining athletic outcomes. Another area of research has looked at cognitive abilities. Some studies have found that women with longer index fingers, indicating higher prenatal estrogen exposure, scored higher on certain cognitive tests at younger ages, particularly in verbal abilities. Interestingly, older women with longer ring fingers performed better than those with longer index fingers on some tests. But again, these are correlations, not causations. They're hints that prenatal hormones might influence cognitive development, but they're far from definitive. Researchers have also looked at personality traits. One study examined finger length ratios and their connection to masculinity and femininity in nearly 1,000 women. They found that women with shorter index fingers, and therefore longer ring fingers relative to their index fingers, were more likely to describe themselves as assertive and competitive. It's a tantalizing suggestion that prenatal hormone exposure might influence in personality development, but we have to be careful here. These studies often have small effect sizes, the correlations are weak, and there are many confounding variables that could explain the relationships. One particularly controversial area of research has looked at sexual orientation. Some studies have suggested that finger length ratios differ between homosexual and heterosexual individuals. The theory is that different prenatal hormone exposure might influence both the developing finger bones and the developing brain structures related to sexual orientation. But this is a highly sensitive topic, and it's important to note that the research is complex and sometimes contradictory. Science is still working to understand these relationships, and we should be cautious about drawing firm conclusions. What's absolutely clear is that many of the popular claims about finger length ratio are complete nonsense. Some people claim that finger length ratio can predict intelligence. There's no scientific basis for that. Some claim it can predict sexual potency or genital size. That's completely unfounded and frankly silly. Some claim it can predict faithfulness in relationships or chivalry or moral character. These are pure fiction. The only legitimate connection is between finger length ratio and prenatal hormone exposure. Everything else flows from that fundamental relationship. Here's what's important to understand. Correlation is not causation. Even when researchers find a genuine statistical correlation between finger length ratio and some other trait, it doesn't mean that the finger length is causing the trait. It means that both the finger length and the trait might be influenced by the same underlying factor. Prenatal hormone exposure. The finger length is a proxy, a marker, a biomarker. It's a window into the hormonal environment you experienced in the womb. It's not the finger length itself that matters. It's what the finger length tells us about your early biological development. This is actually why scientists find finger length ratio so valuable as a research tool. You can measure it easily and non-invasively. You don't need blood tests or brain scans. You just need a ruler and a hand. It's cheap, it's simple, it's objective. And it provides information about prenatal conditions that happened decades ago. Once you're born, measuring hormone levels in your blood doesn't tell you what you were exposed to in the womb. But your finger length does. It's like a biological record of your fetal past. The scientific validity of finger length ratio has led to over 300 research papers being published in the last 10 to 15 years alone. Researchers have investigated connections to autism, attention deficit disorder, various cancers, cardiovascular health, fertility, and dozens of other conditions and traits. Some of these connections are stronger than others. Some are still speculative. But the fact that so many researchers are investigating these relationships speaks to how seriously science takes this measurement. One particularly interesting area of ongoing research is the connection to disease risk and disease susceptibility. Some studies have suggested that finger-length ratio might be statistically associated with risk for various types of cancer, including prostate cancer, testicular cancer, breast cancer, and cervical cancer. The underlying theory is that these hormonally influenced cancers might be influenced by early hormone exposure. And since finger-length ratio is a reliable marker of prenatal hormone exposure patterns, there might be a meaningful biological connection. But we absolutely need to be crystal clear about this. Having a longer ring finger does not mean you will inevitably get cancer. These are purely population-level statistical associations and correlations. They are not deterministic. They don't determine individual outcomes for any specific person. Similarly, some research has looked at whether finger-length ratio might predict fertility or reproductive health. The logic is similar. If prenatal hormones influence reproductive development, and if finger-length ratio marks prenatal hormone exposure, then perhaps there's a connection. Some studies have found associations with sperm count and with family size. But again, these are weak correlations based on population statistics. They're interesting scientifically, but they're not predictive on an individual level. The genetic research has also revealed something interesting about sex differences. Men, on average, do have lower 2D to 4D ratios than women. That's consistent with the hormone exposure theory. But there's enormous overlap. Many women have ratios lower than many men. This variation within sexes is actually what makes the genetic research so interesting. If the trait were completely determined by sex, it would be boring scientifically. But because there's this tremendous variation within each sex, it means there are interesting genetic factors at play beyond just the sex-determining chromosomes. Researchers have also looked at whether finger-length ratio changes over time. The answer is fascinating. It doesn't change much. Your finger-length ratio is largely set by early childhood. By the time you're 7 or 8 years old, your finger-length ratio is pretty much what it's going to be. It's stable throughout life. This stability makes sense given the theory. Your finger lengths were determined during fetal development and early childhood by genetic and early environmental factors. Once that's set, it stays set. Your bones don't change length significantly in adulthood. This stability is actually one of the things that supports the biological validity of the measurement. If finger-length ratio were a meaningless or random trait, we'd expect to see more variation over time. But because it's stable, it suggests it's measuring something real and fundamental about your biology. The practical applications of this research are still emerging. Genetic testing companies like 23andMe and Ancestry DNA have started offering information about finger-length ratio as part of their trait reports. They can look at your DNA and tell you whether your genetic variants suggest your index finger would be longer, your ring finger would be longer, or whether they'd be about equal. It's not a perfect prediction. Remember, only about 60% of the variation is genetic, but it's interesting. The genes identified in these studies also provide insight into fundamental biological processes. The SMOC1 gene, for instance, has been implicated in bone growth and development. Understanding how genetic variants in SMOC1 affect finger length might teach us something about bone development. More broadly, it might have implications for understanding osteoporosis or other bone disorders. The LIN28B gene is involved in stem cell development, and understanding how variants influence finger length might have implications for cancer research and stem cell biology. What's remarkable about this research is how it demonstrates the power of modern genomics. 100 years ago, scientists noticed that men and women had different finger length ratios, but they couldn't do anything with that observation. With the tools we have now, the ability to sequence millions of people's genomes and perform massive statistical analyses, we can identify the specific genetic variants responsible. We can understand the mechanism. We can replicate findings in independent populations. This is the modern face of genetics. It's not about single genes determining single traits anymore. It's about understanding the complex interplay of multiple genes, each with small effects, combining to create variation in a trait. It's about understanding how environmental factors interact with genetic predisposition. It's about applying big data and statistical power to uncover biological truths that would have seemed impossible to understand just a few decades ago. The 2018 study by Warrington and colleagues represented the culmination of decades of work by hundreds of researchers. It brought together data from multiple countries, multiple populations, multiple studies. It used cutting-edge statistical methods. And it provided definitive answers to questions that had been puzzling scientists for generations. But it's also just the beginning. There are still mysteries to solve. Why exactly do some genetic variants near SMOC1 increase finger length ratio while others decrease it? What's the mechanism? How do testosterone and estrogen actually regulate the expression of SMLCA1? How do the different genetic variants in different genes combine to produce the final outcome? These questions are still being investigated. There's also been interesting research recently looking at finger length in evolutionary context. Some researchers have investigated whether finger length ratios might vary across different populations or ethnicities. The research suggests there might be some variation, but it's subtle and not well understood yet. This is an area where more research is needed. As we move forward, we're going to see more applications of this knowledge. Personalized medicine is coming, where doctors might look at your genetic variants and your biomarkers to tailor health recommendations specifically to you. Understanding your prenatal hormone exposure through measurements like finger length ratio might become part of a more complete picture of your biological risk factors and strengths. What's truly astounding when you step back and think about it is that something as simple as the length of your fingers holds information about something that happened before you were born. Your hands are like a record, written in bone and tissue of the hormonal environment your body experienced in the womb. That's profound. That's remarkable. That's the kind of thing that makes you realize how much more we still have to learn about ourselves. And that brings us back to where we started. Your hands have been keeping a secret. 2 simple fingers hold the key to understanding your biology at a deep level. Science has finally cracked that code. We know which genes are involved. We understand the basic mechanism. We know what it tells us about prenatal hormone exposure. But here's the most important thing to take away. This is just one small piece of who you are. Your finger length ratio is influenced by your genes and your prenatal environment. It might correlate with some other traits. It might tell us something about your biological predisposition. But it doesn't determine your destiny. It doesn't define your capabilities. It doesn't limit your potential. It's simply one data point among millions in the complex story of human biology. We are the products of our genes, yes, but we're also the products of our environment, our choices, our experiences, and our efforts. Genetics loads the gun, but environment pulls the trigger, and sometimes we pull the trigger ourselves through sheer force of will and determination. The study of finger length ratio teaches us something valuable about science itself. It shows us how patient investigation can uncover mysteries, how the combination of observation, hypothesis, experimentation, and technology can reveal truth, how something that seems trivial, the length of your fingers, can actually reveal something profound about your biology. It also teaches us humility. For over 100 years, scientists looked at this trait and wondered about it. They had theories, they made guesses, but they couldn't know the answer until the technology caught up with the questions. That's true for so much in science. There are probably dozens of mysteries in human biology right now that we're looking at and wondering about, not knowing that the answer is just around the corner, waiting for the right technology or the right insight to reveal it. So the next time you look at your hands, take a moment to really see them. Notice the length of your fingers. Think about the fact that this measurement is genetic, determined by variants in multiple genes across your genome. Think about the fact that it's connected to your prenatal hormone exposure, to the biological environment you experienced before you were even born. Think about the fact that thousands of researchers across the world have studied this simple physical trait to understand it better. Your hands are more interesting than you thought. Your fingers carry more information than you realized. And science is still uncovering the stories they tell. In years to come, we might discover even more connections between finger length ratio and other aspects of health and biology. We might find new genes involved. We might understand the mechanisms even more clearly. That's the beauty of science. There's always more to discover. There are always new mysteries to solve. And every time we solve one mystery, we uncover new questions. The journey of understanding ourselves never truly ends. And for those of us who are curious about how the world works and how we work as biological entities, that's the most exciting thing of all. The secret your hands have been keeping is just the beginning of understanding the remarkable biological system that makes you who you are.